and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to Dr. Maryse Bourgault and Jessica Williams about root systems and drought tolerance. Roots are an important factor in drought resistance as they are the primary way by which plants uptake the water they need to survive. However, measuring roots can be a difficult, time-consuming, and destructive process. In this episode, we discuss Maryse and Jessica's work to better understand how drought, root systems, and other traits relate by partnering across studies in greenhouses in the field to study traits that get to the root of drought resistance. We'll talk more about that in a minute, but before we dive in, we want to thank our sponsor, Meter Group. Meter specializes in robust soil moisture sensing, innovative weather monitoring, cloud data logging, advanced data visualization software, and more. Their well-published scientific instrumentation is used worldwide in universities, research and testing labs, government agencies, agriculture, and industrial applications. Listen to their new podcast, We Measure the World, to hear how innovative researchers leverage environmental data to make our world a better and more sustainable place at metergroup.com/fieldlabearth. I'm your host, Abby Morrison. Let's talk about science. Hi, everyone. Welcome to the show. Today we have Jessica and Maryse with us. Jessica is from Golden, Colorado, and received her bachelor's degree in biology from the University of Denver. After college, she worked as a research assistant in 2 fruit fly labs where she learned a lot about genetics before following her real passion into agriculture. Now she is a PhD candidate in plant science at Montana State University. Dr. Maryse is an assistant professor and the new Western Grain Research Foundation Chair in Integrated Agronomy and a joint appointment between the Departments of Plant and Soil Sciences at the University of Saskatchewan. Prior to returning to Canada, she was an assistant professor at the Northern Agricultural Research Center at Montana State University in cropping systems and agronomy. She also spent 9 years in Australia, working mostly in climate change-related research. She also worked as an extension agronomist while living in Queensland. As a crop physiologist by training, PhD awarded from McGill University in 2009, Maryse uses agronomic research, crop physiology methodologies, and modeling to investigate questions of sustainability and resilience in semi-arid cropping systems. Hi, Jessica. Hi, Maryse. How are you doing today?
Very good, thank you.
Good. Hi, Abby.
So good to have you both on the show today. So we are talking about stay green and drought conditions and root systems and semi-arid conditions and how those all kind of intertwine today. So to get us started, can you just kind of lay out the problem you were dealing with your research and how all of these different pieces kind of fit together into a larger picture?
Sure. Yeah. We're talking about 2 projects here today. One that was focused on wheat and barley, and another on peas and lentils. But both have to do with trying to optimize these crops to be able to withstand drought conditions. We were based in the northern Great Plains region, so we were, in the drought conditions that are specific to that area. And we specifically were interested in how the root systems of these crops could come into play in helping them tolerate drought.
Okay. And just for people who are not familiar with stay green as a concept, can you just explain a little about what that is?
Yeah. So stay green is a trait that is a naturally occurring trait that's been observed in several cereal species, so in corn and sorghum and rice, and also in wheat and barley. And the stay green trait, as the name suggests, it just means that these plants stay green for more of the growing season. So they tend to reach maturity where the plants start turning yellow later in the season, and we found that they also tend to enter the grain filling phase earlier in the season. So they produce their seed heads earlier on, and the period of time that we refer to as grain fill, where those seed heads are filling up with starch, that period lasts longer in these plants that have the stay green trait.
Okay. So Like you said, you were kind of researching how all of these things interrelate, to which you had 4 different research questions in this project, kind of mirrored in both projects, actually. So can you tell me what those 4 research questions were?
Yes, so we wanted to see if this stay green trait that we were interested in as a drought tolerance trait, if it might be connected to root systems, if there's something different about the roots of those stay green plants. And we also wanted to just look at how roots connect to the performance of the crop above ground. Also, we wanted to see if there are connections between root systems of plants growing in the field and root systems in seedlings and younger plants in some greenhouse studies, because it's a lot easier to measure roots in a greenhouse study than out in the field. So we're kind of looking for proxies there. And then in my project, the 4th research question was just, are there differences for these root traits that we're studying between wheat and barley? Because those were the 2 crops we were looking at. And then I think Maryse had some additional questions specific to pea and lentil.
Yeah, so with peas and lentils, we have much less information about the kind of traits that might be helpful or related to adaptation to semi-arid areas. So we don't know, for example, we don't have the equivalent of a stay green comparison to do in peas and lentils. So we went a little bit wider in looking at the different genetic material that's out there that has been released by different breeders. And our idea was to try and look at the diversity that might be there and what that looks like in terms of root growth. And partly is because roots are difficult to see, so we have much less information about them than we do for above ground traits. But at the same time, we know for sure that roots must be important in terms of adaptation to semi-arid areas or drought or drier conditions, because they are where the plants actually take up water, and so that's where we were trying to look at and see if some of the results that we've seen in cereals also align with what we're seeing in pulses.
Yeah, I really liked, I was reviewing your paper earlier in preparation for this call, and I liked that it was kind of just research that's taking more into consideration than just the yield. It's kind of looking at, all of the plant features and seeing which ones connect to those qualities and traits that we are hoping to build into the crops that we're breeding. So I thought that was really interesting, and I'm excited to see how that turned out. I kind of know because I read your paper, but yes. I'm excited to share with the listeners.
Yes, and in many ways, we kind of have to do that because yields are so variable and they're impacted by so many factors that actually looking at some of those traits that are, what we call heritable, so traits that are a little bit more stable from year to year, knowing that we have these traits in some of those populations, sometimes it's easier to bet on these traits actually helping yields than just looking at yield by itself and hoping for the best.
Yeah. I really like that. So pleased. So then moving on, now that we know what we were investigating, and by we, I mean you, it's not involved. Can you tell me about how you went about testing these? And Jessica, maybe we'll start with you with the barley and wheat, and then back to Maryse for pea and lentil.
Yeah, great. So I was doing research in 2 different places, if you will, out in the field and also in the greenhouse. So I had 3 seasons of field studies where on the surface it was kind of your usual field trial where I was growing these different varieties of wheat and barley, some of which were stay green and some of which were not stay green, and measuring things on the above ground parts of the plants like yield and things about grain quality and plant height, leaf area, stuff like that. And then in those same field studies, I was looking at what's going on below ground using tools called mini rhizotrons. And a mini rhizotron is basically just a big, long, clear acrylic tube. And we bury these in the ground at a 45-degree angle right after planting the seeds of all of our different plots. And then we have this special scanner that it's basically like a desktop scanner, except that it spins in a circle and it's the right size to be inserted in these tubes. So at any point in the season, I can come along, put the scanner into the tube, and it will capture an image of whatever is coming into contact with the tube, which is soil and roots and the occasional bug or something. So that's how I looked at the below ground portion of things in the field. And then in the greenhouse, I did 2 seedling assays, one that I like to call root roll-ups. And for that, I basically had big sheets of germination paper and line up some seeds along the top, roll up the paper, and then, just keep them upright in a bucket of water. And the roots would grow down in between the layers of those paper rolls, so that in about 3 weeks I could unroll them and measure different traits on the roots of these very young seedlings. And then to look at a little bit older seedlings, I did a project in the greenhouse using PVC pipe that was just cut in half and filled with this stuff called turfus, that it's just like chunks of clay, and it makes it really easy to grow a plant and then wash the turfus off of the roots. If anyone's ever washed soil off of roots, they know that that's a bit of a pain, so the turfus was really nice to have for that. And then I could measure root traits on plants that were more at like the 5-leaf stage, whereas in the root roll-ups I was looking at the 1-leaf stage. And then just kind of was making connections between all of the traits I measured, the ones in the greenhouse, the root traits in the field, and also the above ground traits on the plants in the field.
And Maryse, how did your research go?
Yeah, we use the same system. So we also have these tubes inserted pretty much right after planting. So we go into the field, and it's a little bit of it's a challenge. It's a challenge sometimes, especially if we have some dry soil at depth. So it's a challenge to put them in, but it's really a system that's really neat in a sense that we can go back time after time, and so we don't. This isn't a destructive sampling, and we're not punching holes or destroying the plot trying to look at these roots. So it's a neat system in the sense that we can do repeated measures on the same plots and look at root development through time.
Sure. And how many of these mini rhizotrons do you have, like, around, say, like one plant? Like, is it just hopefully the roots grow around the tube, and if they accidentally grow in another direction, too bad? I mean, how does that work?
It is a sampling, it's around the plant, right? So the mini rhizotrons themselves are a meter long, and because we're going at a different angle, we probably are looking at more than one plant. So because we're looking at a field settings, obviously we'll have, we had crops on 12 inches row spacing. So it does a little bit underestimate the amount of roots that you might have near the surface, but we've also found, and we were concerned at some point that maybe it would overestimate the number of roots at depth, because we were thinking maybe once the root touches the tube, then it's going to be easy for it just to keep going down. But we're not actually seeing that. We're actually seeing roots that come in, touch, but then they might pull away from the tube, possibly because there's no nutrients there, right? So it does take soil for the roots to actually do something. So.
Oh, interesting. Yeah, I think I would have just assumed that they would have been like, oh, this is a nice smooth surface for me to like grow around and grab onto. But, it's not like water where
No,
it's just following the easy path.
Exactly. It's plastic, so there's absolutely nothing for them to to take anything. So
yeah, they don't want to hang out. Hi everyone, I hope you're enjoying the show. Interested in learning more? The two papers featured in today's episode, Relationships Between Roots, the State Green Phenotype, and Agronomic Performance in Barley and Wheat Grown in Semi-Arid Conditions, and Genotypic Variability and Root Length in Pea and Lentil cultivars in a semi-arid environment based on mini rhizotron image capture, are always freely available. They're also part of the open access special section, Belowground Phenotyping, from the Plant Phenome Journal. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Thanks again also to our sponsor, Meter Group. Meter specializes in robust soil moisture sensing, innovative weather monitoring, cloud data logging, advanced data visualization software, and more. Their well-published scientific instrumentation is used worldwide in universities, research and testing labs, government agencies, agriculture, and industrial applications. Listen to their new podcast, We Measure the World, to hear how innovative researchers leverage environmental data to make our world a better and more sustainable place at metergroup.com/fieldlabearth. Thank you for being our sponsor. Let's get back to the show. Oh man, I love so much about this because for one thing, like mini rhizotron is such a cool name for like anything to exist. It sounds like some kind of crazy robot. But I will stop gushing so we can move on to the results. So what did you find?
Yeah, so for the Stay Green wheat and barley, we did indeed see differences between wheat and barley. For barley, the Stay Green lines tended to have more of their overall root length deeper in the soil. And we did the root measurements in the field at the beginning and the end of that grain fill period I was talking about. And the Stay Green barley lines seem to have an increase or lesser decrease in root length over the course of that grain fill period compared to the non-Stay Green lines. For wheat, there wasn't a whole lot of difference in roots in the field between Stay Green and non-Stay Green. So there might be something different going on with Stay Green wheat plants versus barley. However, we did see connections between above ground and below ground, kind of ignoring the Stay Green piece for a minute, and thinking about that second research question, just how do roots connect to above ground plant performance. The increase in root length from during that grain fill period and the greater deep root length in barley was connected to higher yield, whereas in wheat, having more root length overall, just kind of a larger root system, that was what was most strongly connected to having greater yield, grain yield. And we also saw some connections between the roots in the greenhouse and the roots in the field, but something interesting there was that wheat and barley were again different. For instance, one of the traits that we measured was the angle between the outer roots coming out of the seed in that really young seedling assay, the root roll-ups. And we thought that maybe having a narrower angle would be connected to having more deep root length in the field, because maybe the roots would come out and just like go straight down, and then you'd have more roots deeper down in the field. But in barley, we saw that a wider root angle was connected to more deep root length, whereas in wheat, it was more like what we expected, a narrower root angle connected to more deep root length. So, I think that the shapes of the root systems between wheat and barley might be pretty different. Like maybe barley goes out and then comes back in and then does more branching down deep to achieve all of that deep rooting, something like that more. So those were the things we found, and are some of the things leading to more questions.
That's the game with science.
Yeah.
There's always more to learn. Maryse, what did you find?
Yes, well, we also found, slightly different results than what we expected. So in many cases, we assumed that to be successful in a semi-arid environment, in a dry environment, the root system needs to be a little bit lean but go deep. And we didn't really find that with our peas and our lentils. So the peas a little bit more. Jessica's mentioned that, between in that grain filling period, sometimes some of the stay green had a little bit more of that root growth compared to the other ones. And in cereals, we tend to see them kind of peaking at enthesis and then going down a little bit. Whereas with pulses, we don't see that. They just kind of keep making roots, and they somehow, they keep making that. And for peas, we're thinking that perhaps, the cultivars, that if they're investing in roots, then they don't have the energy and the carbon to invest in yield formation. So in peas, that kind of looks like that might happen. So our better yielding cultivars were usually the ones that were in the middle. So they weren't very deep, they weren't very big, but they, at the same time, they were not very small either. So there might be a way here to have some peas that have, a good root system, perhaps at flowering, and then perhaps, trying to push them into a direction where they would stop making those roots, a little bit like cereals would. But then by contrast with lentils, the bigger root system, the better it was. And it really didn't matter if those roots were nearer the surface or below 30 centimeters. The more they had, the better it was. And we're also thinking that lentils is a small plant to start with. And some of the breeding objectives has been to try and make bigger plants, taller plants, so it's easier for the combine to harvest them and harvest those lower pods. So if we're pushing the plant to become bigger, then perhaps it makes sense that we need a bigger root system to feed it as well.
Interesting. I love all the differences between all of these results.
Yes, and it's, we tend to have this thing, that like, well, this is the ideal root system and perhaps it's going to work for every crop out there, but it doesn't seem like that's the case. Like we probably need to be a little bit more specific with each of the crops that we're looking at.
Yeah. Oh man. And I'm just thinking, like, even studying like different ideal root systems, across like different environments, like obviously that's, well, maybe not obviously, but I would assume that would be really different too.
No, that's very true.
Yeah.
So yeah, it seems like there is a ton of room for further research, to which my next question is all about kind of where we are going with this research. Obviously, it seems like there were some surprises in your research that has, like Jessica said, led to more questions. So what are some of the future research questions that you think would be worth investigating?
Yeah, absolutely. What you said about the ideal root system being different for different environments, I think is pretty important. The stay green barley results that I saw with having this deeper root system that continues growing during grain fill, I think that might be really beneficial in an environment where you get a lot of rainfall in the spring earlier in the season. You have a deep soil profiles that can hold and store that water down deep, and then later in the season it gets very dry. That's the type of environment we were working in. And so having that, those roots continuing to be active deep in the soil, that would be beneficial. But if you don't have those deep soils, and if you do have rainfall later in the season, then maybe more shallow root length would be better. So something we would like to do is test out these stay green root characteristics in different environments. So we're doing some genetic mapping to zero in on the genes behind these, or the genetic areas behind these stay green root traits, so that then we can make some very specific lines of barley to then be able to do replicated studies in different environments and test out like different irrigation strategies and look at the roots and all of that to see how these traits interact with the environment more and where they might be beneficial or not so beneficial.
Sure. Maryse?
Yes, we do have a project currently that we are actually using the same material that we've used in the field to look at another trait, so another drought adaptive trait. And part of our hypothesis there is that some of these traits might actually be co-located, or they might be linked in a mechanistic physiological way. So it might be that having those deeper roots allow some other traits to happen. So for example, we're looking at the accumulation of sugars in stems, and right now we know that peas have a lot more than even wheat, in barley, in lentils. So it's going to be really interesting to see if there are differences between the cultivars that we have, and if these differences actually map with the differences that we see in root growth. And they, ideally they would be independent, and then we could provide that to breeders and say, well, both of these things are good. So if you combine them, then hopefully you get even more of that benefit and that drought tolerance and that adaptation. We also have a proposal, again, like Jessica said, about looking at genetic mapping of those populations. So we have a proposal to start making some of these populations in peas and lentils, so that we can look at things like that, so that we can grow them, try and look at maybe 300 lines. Like that becomes a little bit more intensive, because that's something that's really difficult to do in the field. It's really almost impossible, because with the mini Rhizotron, the maximum amount of plants that we can really do in a day is maybe 24, maybe 30 if we push it, and we have dedicated students that are willing to do 12 hours out in the field. But again, weather happens as well, so it's not like you can count on having 10 days of perfect weather where you can actually go and do 300 lines. So we're trying to test different systems as well to see what we can do in the greenhouse that would actually relate well to what we're seeing in the field.
Sure. Bountiful opportunities for further research for both of you. That's wonderful. I have a slightly more philosophical bonus question for you. Maybe not as philosophical when applied to plants as it is to humans, but what is like the nature versus nurture aspect of this? Like, you are talking about these ideal, root systems in an environment, but how do we know how much flexibility plants have as far as that? Like, is it something where normally this species of plant or cultivar grows roots that have this shape or length, but now because it's really dry, they're reaching deeper or farther out? Or is it, I would assume that there's a certain amount of genetic limitation on how far, it's not like they're going to be able to bury to the core of the earth, but what is that line typically? Like, how flexible are plants when it comes to this kind of thing? Or do we just not know yet?
Do you want me to take that one, Jessica, or you want to take it?
Well, I'll just say a little about the genetic, because I've been working on some genetic mapping already of these stay green root traits, and in those types of projects, we're always looking at how much of what we observe is due to genetics and how much of it is due to the environment and how much of it is due to the interaction between the genetics and the environment. And with roots, I think Maryse, would you agree that it's safe to say that with roots, the environment component is usually bigger?
I don't know if it's bigger, but it's certainly important. It's certainly important, but to me, it's not so much how big it is compared to the genetics, but is it more is the genetic component more present looking at the roots versus looking at yield, because that's the two indicators that we have, to look at productivity and look at adaptation into these systems.
Yeah. So I think that's part of why we're excited to do this. To do the genetic mapping, we need to use a really large number of lines. So it's definitely not practical to do many rhizotrons on a genetic mapping population. But that's a first step to developing some other lines that will have like very specific genetic combinations that we're interested in, and that's a much smaller number of lines, and then we can test them in lots of different environments and do things like try out different irrigation amounts and timings of water application, maybe look at different fertilizer rates, because in agriculture a huge part of the environment is the management as well. So we definitely want to try to explore how that comes into play with these root characteristics that we're looking at.
Yeah, I might just add that we knew that was definitely a risk in starting this experiment, that perhaps there was just so much flexibility in root systems with pulse crops or with cereal crops that perhaps we wouldn't see differences between the different lines. What encourages me is that even though we do see that flexibility between years, and of course, water and nutrients, all of these will have an impact on whether or not the crop needs to go, or does it grow? Does it need more of that water? Does it need more of those nutrients? What encourages me is that we're seeing cultivars rank more or less the same from year to year. So even though you do see a lot of that flexibility in that response of root growth to the environment, we're seeing some cultivars have bigger root systems pretty consistently. So even though it's, the absolute amount might be quite different, if we know how they rank to each other and how they compare to each other, and they always or mostly compare similarly between years, then that gives us a little bit more confidence about the genetic differences that we might see between them.
Sure. I just find it an interesting question, because what I love about science and seem to come to the fore in this project specifically is just, the depth of complexity between all of the relationships between all of those things, and how scientists are always working to try and isolate like one thing to test at a time, and then making those connections between them. It's the, like we said at the beginning, is looking at not just the yield, but also the roots, and looking at not just the genetics, but also the environment and also the management. And obviously you can have the best, root genetics in the world. If there's concrete buried underneath that you didn't know about, like, it's not really going to make that big of a difference. So I just wanted to kind of, probe your minds a little bit about that, because I just find it really interesting. So thank you for indulging me with the bonus question. I have 3 questions left for you at this point. So the first one is, if people want to learn more about any of the things that we've been talking about today, where can they learn more?
In the COVID year of 2020, so normally on our research center farms, we do field days where we invite people to come to the farm and learn more about the work we're doing there. But thanks to COVID, we did a virtual field day that year. And so I have a couple of YouTube videos where I talk about this project in one of them, and another one where I talk about the genetic mapping project that kind of follows this one. So I don't know if you can share links with people, but those are out there.
Absolutely. We can add links to that in the show notes. Maryse, anything from you?
Well, I might add that both of these papers are open access, so anyone can actually have a look at those papers if they want to. Our contact information is there as well, so if people want to know more. I realize that scientific papers can be a little bit dry for people sometimes, but we're available to answer questions as well if need be.
Sure. Yeah, both of these papers actually are also part of a special section for the Plant Phenome Journal. We'll have links to that in the show notes as well, but if you're interested in this kind of research, you can find other open access papers to read as well as part of that special section and just really learn up about it. So the second question then is, if people want to take the next step and get involved with this kind of research, what can they do?
Well, I think remember that roots are important.
Yeah, for sure.
Yeah, that's a good question, because at some point we're thinking about maybe getting some citizen science into tracing of these roots, because that's one of the really labor-intensive part of it. So the idea was thrown out there that perhaps we could get people involved, and if they wanted to trace roots while they were commuting from from work to home or something like that. We haven't quite got to that point yet, so and we weren't sure how boring that might be for people. So
it's relaxing.
So if there's an interest out there, I'd love to hear it.
Sure. Yeah, we'll have your contact information in our show notes as well. So if people are interested in root tracing, please reach out. I think it could be relaxing. But I guess I don't know what's involved yet. So
it's like a coloring book, just a boring coloring book.
Perfect. It's like the adult coloring books where it's just more intricate.
It could definitely be meditative and.
There you go. If anyone is interested, please reach out. And then the last question I have for you today is, what is one fun fact that listeners would not know about you if all they had was your research?
Well, I would say, so I live in Bozeman, Montana, which is a pretty fun place to be if you like winter. And one of my number one hobbies is going skiing with my dog.
Aww. I'm going to guess your dog does not have tiny, adorable skis.
No. I go cross-country skiing out in the national forest, and he runs.
Beautiful.
Yeah.
Maryse, what about you?
People would not know this about me, but I'm a big gamer, actually. So I don't tend to reveal that too often, but I play.
Do you have a preference?
Well, I play with my brother, actually. So we get together and we really got into it during the pandemic, but we get into it and play together.
Nice. Are there any specific games that you like the most?
Yes. Are you sure you want me to tell them? This is not an endorsement.
You can if you want to. Yeah. Not an endorsement.
I used to play a lot of World of Warcraft, and now we've switched a little bit more into Elder Scroll on the PlayStation 4.
Oh, nice. Well, wonderful. Those are some super fun facts, and this has been a wonderful conversation about some incredible research. Thank you so much for all that you are doing to advance the science, and thank you so much for your time to be on the show today.
It's been a pleasure. Thank you for having us.
Yes, thanks, Abby. This was fun.
Hi, everyone, and welcome to our Student Spotlight, where we highlight the work of graduate and undergraduate society student members. Today, we'll be talking to Olan Rawaju. Welcome to the show. Can you start us off by introducing yourself and where you're studying?
So my name is Olan Rawaju Edward Adeyemi, and I'm originally from Nigeria. I obtained my Bachelor's of Agriculture at the Federal University of Agriculture, Abeokuta in Nigeria, and I am currently a MS candidate at the Department of Plant Sciences, University of Idaho.
Wonderful. And what are you currently researching?
Yeah, thank you. My current research explores the effects of nitrogen and sulfur fertilizer rates on the nutrient utilization, grain protein, and yield of three barley classes. Those are malt, food, and feed barley. So this is because it is believed that sulfur improves nitrogen uptake and assimilation into plant tissues. So we are trying to see how the interaction of nitrogen and sulfur could bring about improved grain protein and yield for the aforementioned barley classes. So this is also to improve the nitrogen and sulfur fertility recommendations of the University of Idaho, which was last updated in the early 2000s. And this also will provide fertility, nitrogen and sulfur fertility recommendations for Idaho barley growers to improve barley yield and quality while minimizing their nutrient inputs and environmental pollutions, which could arise from misuse or overuse of fertilizers.
Great. And if you could have your dream research project, what would that be?
Yeah, that would be to research how the interaction between effective nutrient management strategies and plant breeding techniques could contribute to meeting the rising global food demand, by improving soil and crop quality and nutrition, and also by developing and recommending better crop varieties that are genotypically and phenotypically resilient and that are also climate resilient. So I am really my dream project would be to see the interaction between nutrient management and plant breeding, like how they can come together to improve crop yield.
Awesome. If you'd like to get in touch with Olanrewaju about his work, we'll have his contact information in our show notes. Thank you so much for being on the show today, and best wishes on your future studies and career. Thank you.
Thank you very much, Abby.
Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.